Hey everyone, good evening. Welcome to today's session. My name is Daniel Menacia, fourth year medical student, MD, loading class of 2025. And today I'm going to be going over Jack Weston Practice Exam 6, Biochemistry Passage 1. And I'm going to be breaking down this passage and answering the questions, sort of like a test taking skills workshop. And hopefully, you're able to leave this session with sort of improved test taking skills, knowing, how to highlight pertinent information, how to filter extraneous information, and how to better tackle these biochemistry questions. All right, so without any further ado, I'm going to start sharing my screen and we can get started. All right, so I'm going to be reading and highlighting as I go along. Okay, so without wasting any more time, several lines of evidence have suggested that there may be a candidate gene promoting carcinogenesis on the P arm of chromosome 10. Okay, so just from this first sentence, we know that we're looking for a candidate gene that's promoting carcinogenesis. Okay, so it's important to know the meanings of words. Throughout your medical career, you're going to be using a lot of words that look like this. So carcinogenesis, development of cancer. So candidate gene promoting the development of cancer on the P arm, the short arm of chromosome 10. That's where it is. Okay. Now, using a restriction fragment length polymorphism approach, extraneous information. Loss of heterozygosity at chromosome arm 10 was detected in several prostate cancers. Okay, so loss of heterozygosity, okay, at chromosome arm 10 was detected. And I guess the sort of disease we're using is prostate cancer. Okay, so loss of heterozygosity is the mutation, okay? That's happening on the P arm, and this candidate gene is losing heterozygosity, basically. And remember, guys, from your cell biology or genetics, what is the opposite end of heterozygosity? Homozygosity. So this candidate gene that's promoting carcinogenesis on the short arm of chromosome 10 is showing signs that it's losing heterozygosity at its gene loci, okay? All right, so now, detected in several prostate cancers. Prostate cancer is what we're using for our case studies. In functional studies, a chromosomal DNA fragment from 10p was able to suppress prostate cancer cell proliferation. Okay, providing additional evidence for the presence of a gene implicated in carcinogenesis. Okay, so they did some functional studies and they showed that chromosomal DNA fragment from 10p was able to suppress prostate cancer cell proliferation. It's giving us some signs and keys here as to what type of gene it is. But we'll find out more as we read along. Okay, so providing additional evidence for the presence of a gene implicated in carcinogenesis. Okay. Excuse me. Cooper-like factors, okay, and you will come across a lot of these factors that you might not know of. When you see them, just take note of them, remain calm. That's the purpose of the test, to test your ability to see new information and be able to apply it immediately. Okay, so Cooper-like factors are a group of transcriptional activators. Okay, so they activate transcription. So remember your central dogma of cell biology. Transcription to translation, and you get your protein. So transcription is your DNA to RNA. Okay, that appear to be involved in different biological processes, including carcinogenesis. Mutations at the KLF6 locus are relatively infrequent, but may be implicated in prostate cancer, what we're studying, what we're talking about in this article, passage, whatever you want to call it, because the absence of KLF6 signaling promotes carcinogenesis. Okay, this is very, very important. So the absence of KLF6 promotes carcinogenesis. So that means it's a what? Tumor suppressor or proto-oncogene. Its absence promotes carcinogenesis. It's a tumor suppressor gene. Okay, if its presence or gain of function mutation promoted carcinogenesis, then it would be a proto-oncogene, which gets converted to an oncogene because of that gain of function mutation. Okay, figure 1 shows the KLF coding sequence and the nucleotide, which is most commonly mutated. Okay, so you see your coding sequence, 5' end, 3' end, and you see the most commonly mutated nucleotide is the guanine. Okay, just take note of that, move on. Because again, for me, the MCAT was almost 7 years ago now. And I don't remember how timed it was or what the timing was, but I remember it was timed. So you got to move fast. Okay. So that was the point I was trying to make with that. So note the information, note the mutated nucleotide, take a look at it, move on. Don't try to, unless a question asks you, don't try to start looking, oh, okay, is there a triple repeat? Is there a start codon? Don't do that. Okay, move on. All right, to clarify the role of KLF6 in prostate cancer, where the KLF6 is the candidate gene, researchers examined the gene for loss of heterozygosity and mutation. Okay. So expression, loss of heterozygosity and mutation and expression change in 96 prostate cancer samples, including 21 xenografts. Okay, so what are they trying to do? They're trying to clarify the role of KLF6 in prostate cancer. Okay, that's what the experiment is trying to do. Okay, and whether KLF6 is the candidate gene that was talked about at the start of the passage, because they're suspecting it. That's what research is about, okay? So the researchers are examining for a loss of heterozygosity, which they said is the primary mutation that occurs on the short arm of chromosome 10. And okay, the rest of this is extraneous information. You know, it's there, but you know, it's not the most high yield points you need to know, okay? Several well-mapped microsatellite markers in or near the KLF6 locus were used to detect the loss of heterozygosity. So we're using microsatellite markers to detect the loss of heterozygosity in prostate cancer, okay? All right, that's kind of the core of the experiment, okay? All right, microsatellite markers near the KLF6 locus to detect the loss of heterozygosity. So I'm guessing, well, I'm not, no need to guess, I'll just read. Xenograft/cell lines by homozygosity mapping of deletion method, HOMO-D. Again, extraneous information. You don't need to memorize or know the methods. I mean, you will have to know some in medical school, but right now, just, you know, read it, move on. So the HOMO-D mapping technique is based on you need to know how it works, okay? Somewhat. They'll give you how it works. Just note it, okay? The HOMO-D mapping technique is based on a mutation occurring in a gene that is heterozygous more than 75% of the time, okay? When 5 or more nearby markers show a homozygous genotype, okay, so heterozygosity less than 0.75, a cell is established as having lost heterozygosity. Okay, so there's a score, right? The cell is established as having lost. So this is like a rubric, like a grading rubric, right? So if a microsatellite marker that is heterozygous more than 75% of the time, okay, it's found to have a heterozygosity score less than 0.75, which is, you know, 75%, then it's become homozygous, okay? And the cell is established as having lost heterozygosity. Okay, so this is how they determine whether a cell has lost heterozygosity. Figure 2 orders the microsatellite markers based on proximity to the terminal ends, okay, based on their proximity. So that's what microsatellite markers are. Non-coding pieces of DNA. If you didn't know that, please hit me up. We can kind of go over some cell biology, biochemistry. Well, it's more cell biology than biochemistry. But still very important to know. So non-coding sequences of DNA, and they're usually close to the terminal ends of the chromosome, closer to the telomeres. Again, that's why they're microsatellites. So based on their proximity to terminal ends of 10p, the short arm of chromosome 10, to which marker 1716 is the most proximal. I don't know why they included that there, but the fact that they're including it means that they want you to know. Okay, so don't spend your time trying to interpret this right away. Just take a quick look at it, note it, and okay, loss of heterozygosity detection at KLF locus using HOMO-D method. Okay, just note it. Okay, if you spend too much time, you're losing exam time. Okay, now we're gonna start answering the questions. So really quick, question 1. Which of the following is the most likely promoted which of the following most likely promoted carcinogenesis in patients from whom the cell lines were taken? So increased apoptotic signaling. Wrong answer. Okay, increased apoptotic signaling will never promote carcinogenesis, ever. Okay, wrong answer. Okay, in case you don't know, apoptosis is programmed cell death. So you have your BAC and your BAX genes, and when they get activated either by increased mitochondrial damage or whatever, like there's factors that cause apoptosis that I can go over at a later time. If you want to talk about it, but that's not the case in this. It's not the answer in this case. Increased KLF6 signaling. This is a trick answer because we already established that KLF6 is a tumor suppressor gene. So increasing KLF6 will never promote carcinogenesis, because we've established it's a tumor suppressor gene because its absence promotes carcinogenesis. So if it was a proto-oncogene, yes, definitely, then this would be the right answer, but it's not. So increased KLF6 signaling, no, because the absence of KLF6 is what promotes carcinogenesis. Okay, so wrong answer. Increased dependence by cancer cells on glucose for energy. Very, very wrong answer. Bad distractor even. We didn't talk about glucose at all in this passage, so no. A mutation to the genome of a somatic cell. Yes, correct. Mutation we're talking about here is loss of heterozygosity or the guanine mutation, I guess, which causes the loss of heterozygosity on the KLF6 gene locus. So that is the correct answer. Yes. Okay, so 75% of people got it right. Great job. All right, moving on.